Hello, everyone. Welcome to our webinar. It's entitled Therapies Targeting the Genetics of Parkinson's Disease. And this is the third of a series of quarterly webinars that are all sponsored by the Cure Parkinson's Trust, Parkinson's Movement, and Journal of Parkinson's Disease. And you will see that we have 4 panelists. I'm chairing this discussion. My name is Patrik Brundin. I work at the Van Andel Research Institute. And I'm, of course, very interested in Parkinson's research. And I also am the editor, one of the 2 editors-in-chief of Journal of Parkinson's Disease. And I'm going to let the panelists introduce themselves. So can we start with you, Ben?
Sure. Hi, everyone. My name is Benjamin Stecher. I am an independent patient advocate. I'm based in Toronto, Ontario. And I've kind of been traveling the world for the past 2.5 years, trying to understand this disease for myself and trying to see what I can do to push things forward as best I can.
Thank you. Sid.
Hello, everyone. My name is Gan-or. I'm a geneticist based in Montreal at the Montreal Neurological Institute. My work is mainly focused on the common genetic forms of Parkinson's disease, GBA, LRRK2, but also generally genetics of Parkinson's disease. I've been working on GBA in the last 12 years, and I'm very happy to participate in this panel.
Thank you. Gaurav.
Hi, everyone. This is Gaurav Gupta. I work with the Silverstein Foundation. We're based in New York City. We were actually founded by a group of drug developers to help bring new therapies to the clinic for GBA Parkinson's, and we're proud to say that we're going to help bring 3 new clinical trials into the clinic in the next year or so. I'm delighted to be participating and look forward to the questions.
Roy.
Hi, thank you for inviting me for this panel. My name is Roy Alcalay. I'm a movement disorder clinician researcher from Columbia University. My research focuses on genetics and biomarkers in Parkinson's disease, specifically GBA and LRRK2.
And then up on my screen, I have another person who goes by the name of Dr. Gaurav Gupta, but he was drinking coffee and doesn't look like him. I'm not sure if somebody has entered our webinar, but you're welcome. You have a pair of spectacles on. Who are you?
Looks like Jonathon Silverstein.
Jonathon. Yeah, that's Jonathon.
Jonathon Silverstein. That is great. Jonathon, welcome. So Jonathon doesn't have the sound on, but I can say that Jonathon is the founder of the Jonathon or the Silverstein Foundation, which focuses on GBA mutations in Parkinson's disease. And he obviously works closely with Gaurav, and I think that's how they ended up logging in on the same portal and have the same name. But you're welcome. If we can get the sound to work on your panel, you can chip in whenever you want to. So what we're going to do today is we're going to go through the 2 of the major genetic risk factors for Parkinson's disease. And we're going to start with GBA and then segue over to LRRK2. And if we have time at the end, we'll also discuss other forms of genetic Parkinson's and how one can target these. But I want to start then with what do we know about GBA mutations and how they contribute to Parkinson's risk, and how large is that increase in risk if one has a GBA mutation? Ziv, do you want to answer this one?
Sure. So the way we look at it, generally we can divide the mutations, the GBA mutations, to several groups. Usually we divide them according to how they affect Gaucher's disease. So we have severe mutations, so these mutations will cause the severe types of Gaucher disease, type 2 and 3. The mild mutations are those that will cause the mild form of Gaucher's disease, type 1. And we also have polymorphisms that do not cause Gaucher disease, but do affect the risk for Parkinson's disease. So these 3 groups have a differential effects on the risk for Parkinson's disease. So those with the common polymorphism, the 2 that we are usually talking about are E326K and T369M.
Could you just explain for the lay audience what you mean by these weird code names?
Yeah. So these are names of variants in the genes in the GBA gene. They are not very rare, about between 1 to 2% of the general European population carry them, but they have a very mild risk effect. So we call it an odds ratio, and the effect is around 2, which means that if the general population, if we all have about 1% chance to have Parkinson's disease, so patients who carry these, individuals who carry these variants, they have double the risk, which is 2%. So they have 98% chance to never have Parkinson's. So the risk is small. And then we have the mild GBA mutations, which also have quite small effects, usually between 2 to 5, depends on the studies. And we have the severe GBA mutations. They are a bit more difficult to quantify because they are more rare, but based on the studies that we do have, they increase the risk by 10 to 20-fold, I would say.
So this is much more complicated than the genetics we learn in school, where we talk about one mutation, perhaps one disease. We've already described there are different mutations and they increase the risk for the disease in a very variable amount. And I guess it makes it complicated to understand the biology of GBA.
Right, it does, and these different mutations, they have different effects on the protein. Some of them create no protein at all. There's null mutations, and there are mutations that will cause the protein to be retained in the endoplasmic reticulum in a specific part of the cell, and they don't reach the lysosome where the protein is supposed to work. Each mutation causes different things, so this makes things much more complicated.
So if anybody has questions, I'm now speaking to everybody listening in, and actually we have over 100 people listening in today. You're welcome to ask the questions. There's a little box in the webinar where you can pose them, and they'll pop up on my screen, and we'll try to work through as many of the questions as possible. And one question might be, what is the lysosome, and how is this relevant to Parkinson's disease? What does the protein that GBA makes do in the lysosome?
So the lysosome is a part of the cell that's responsible for degrading proteins and lipids, even viruses. So you can think of it as a factory for breakdown of materials. And the role of GBA in the lysosome is to degrade specific types of lipids that are found in the membrane of the lysosomes. These lipids are called glucocerebrosidase. It doesn't really matter the name, but GBA breaks them to glucose and ceramide. So that's what GBA normally does.
Now, if somebody knows that they have a mutation, is there anything they can do? Is there anybody on the panel who would like to address this? So, of course, we know at the outset there is no therapy currently that will stop Parkinson's disease or slow its progression. That's a sort of holy grail of the research. But if one knows that one has a mutation, is there anything one can consider regarding lifestyle or diet?
So I can try and address that. So I have one of the mutations myself, the N370S mutation. And as far as we know, there isn't really anything that you can do from a lifestyle perspective beyond what normal people with Parkinson's disease can do, which is to make sure you exercise as vigorously as possible and to make sure you eat or you live as healthy a lifestyle as you possibly can. However, there are some ongoing trials that look pretty promising that you can try to enroll in and that could provide some benefit. And there's about 3 major ones that I think we'll be discussing to some degree later on, as well as some others that might be coming online in the future thanks to the work of the Silverstein Foundation as well, and a few others.
So that's a very good answer. And of course, it's an absolutely important question that everybody who has a gene will ask. And of course, we wish that in the not too distant future, there'll be a better answer we can give where there is something one can do that we know reduces the risk. Somebody's actually already asked a question. It's somebody called Jose Lanciego. He asked, to what extent are GBA mutations associated with other synucleinopathies like dementia with Lewy bodies or multiple system atrophy? And I don't know, Roy, do you want to answer that question? I think that's a great question.
Sure, it's a great question, and I think it, so just to take a step back, Parkinson's disease is considered a synucleinopathy because the most abundant protein in the brains of people with Parkinson's who donated their brains to research is alpha-synuclein. The same protein is also deposited in people with a sister disease to Parkinson's that is more presents with more cognitive symptoms and less motor symptoms, and that's Lewy body disease or dementia with Lewy bodies. And the association between GBA and dementia with Lewy bodies is actually even stronger than the association between GBA mutations and Parkinson's. Another disease that has alpha-synuclein deposits is multiple system atrophy. A few studies from different places didn't identify an association between the diseases. In our brain bank at Columbia University, we absolutely saw an association, and one of our MSA autopsies actually is of a patient who had Gaucher disease, so 2 GBA mutations, and didn't know about it. MSA is a rarer disease, so the association is harder to prove or disprove, but people are working on it. Lastly, the most common disease where we see alpha-synuclein deposits in the brain is Alzheimer's disease. So also in Alzheimer's patients, roughly 1/3 would have alpha-synuclein deposits in the brain, and whether these are associated with GBA or not is yet to be studied. Yet we don't really know yet.
So that's a great answer. Siv, I think you wanted to add something to this?
Yes, so in recent years, we found that there is a prodromal condition for the synucleinopathies that Dr. Alcalay just mentioned, which is REM sleep behavior disorder. So people with this sleep disorder, they will enact their dreams. The motor muscles are not paralyzed as they should be during REM sleep, so they enact their dreams. And people who have that and who are truly diagnosed with that in a sleep lab, they are going to either develop Parkinson's disease or dementia with Lewy bodies. Most of them, and a small minority of them, develop multiple system atrophy. And the interesting thing is that and not very surprisingly, is that GBA mutations are very common in this group of individuals. So that's an interesting early prodromal marker for development of overt synucleinopathy that is relevant for carriers of GBA mutations.
So could you say a little bit about what the risk is of developing a neurodegenerative disease if it is a clinically verified REM sleep behavior disorder?
So the risk is very high. It's currently, we would say it's above 80%. And the only reason we don't say 100% is because when we do these studies, we currently have a big study of almost 1,300 patients that are being followed up. It's going to be published very soon. So when you follow them up for more than 15 years, almost 90% of them develop either Parkinson's or dementia with Lewy bodies or multiple system atrophy, and the rest were lost to follow up. So that's why I cannot say 100%. But so people with this REM sleep behavior disorder, what they don't have a sleep disorder, they have a prodromal synucleinopathy. That's the right way to look at it. It's just a prodromal symptom that is very indicative that they're going to develop an overt synucleinopathy in the future.
So to summarize, before we go on to the treatment part, it seems then like GBA mutations, there are several of them, they alter the protein's function in a way that we don't fully understand, and they can cause a variety of neurodegenerative diseases, not just Parkinson's. And I think we should move on then to the kind of core of our discussion today. Can one target GBA with experimental treatments? There isn't an established treatment yet, and I don't know, Gaurav, do you want to talk about that? The Silverstein Foundation is, of course, devoted to this to a great extent, and would you just give us an overview of areas, and others can chip in, what kind of therapeutic approaches there are?
Thanks, Patrik. I'd be delighted. So I think the way to think about this is how can we try to target the disease? And so one option is with small molecules or a drug you would take by mouth, like a pill. Another might be with antibodies, which is like a shot you get at the doctor's office. And then we can talk about more exotic types of things like gene therapy, cell therapy, or RNA therapy. So currently, there's actually, as Ben referenced, a few trials ongoing, 2 sponsored by big pharma, which is very exciting. All 3 of these trials that I think are in the clinic right now focus on therapies you take via pill to help try to improve the function of the GBA enzyme. 2 of them work to try to improve the function. One of them works to try to reduce some of the buildup of the problematic lipid that might happen if you have dysfunctional GBA. So that's kind of what's in the clinic now, is these pills that work on trying to change the enzyme's function or the sequela of the enzyme's function. And then in more broad strokes, and we're happy to dive into more of that, in more broad strokes, because it is a genetic disease, we can start to think about things like genetic medicines. And so one thing that I think is very exciting is the development of gene therapy, in which we can actually replace the gene through the delivery of a viral vector. And then down the line, there may be more exciting things such as protein degraders and other things that might be ultimately relevant.
Do you think that any of these therapies would improve symptoms, or is the aim to slow progression of the disease?
It's a great question. Since we have to assume that at least some of the symptoms are due to the gene's function itself, there probably would be an improvement in symptoms if you really could target the gene effectively. But realistically, Parkinson's disease symptoms are actually very well treated by a variety of medications that are on the market today, which actually makes it sometimes hard to study the progression of the disease. Really, we believe, and what's exciting about having a group of patients that have a single genetic mutation that increases the risk for the disease, what's exciting is that by targeting that mutation, we might actually be able to modify the disease progression. So the goal of all these therapies, Patrik, to your question, is to change the course of the disease, not just kind of plaster it over by fixing the tremor or the dyskinesia or something.
So if one addresses a disease where there is a defined mutation, is there a good chance that there could be biomarkers that one could follow? And by this, I mean things that you can measure in fluids, in blood, saliva, spinal fluid, that will reflect whether the therapy is working or not. And I should say, while we think it's great that neurodegenerative diseases progress slowly, it actually makes it very hard to test new therapies because you have to follow patients for a long time before you can see if the therapy is having an effect. So anybody want to chip in, Gaurav, or anyone else, talk about the possibility of GBA-related biomarkers, and what would they be?
I can make a brief comment and then I'll let my colleagues jump in. I think broadly, yes, and I think there's two categories. One is biomarkers you could pull from someone's blood or perhaps their spinal fluid. And the second category would be something you could do via like an imaging test, like a PET scan or something. All of those, I would say, are in development and potentially very, very exciting and very useful tools. But I'll pause there and I'll let some of my other colleagues maybe dive into some of the specifics since we have other experts on the line.
Roy, do you want to have a go at this question?
Sure. So to just start with the previous question about intervention targeting GBA, I think we are in exciting times in Parkinson's research, and I really, really hope that in 5-10 years, people will look back at 2018 and say, we don't believe that that's how we treated Parkinson's 10 years ago.
Yeah. Could you just give us the historical perspective? GBA mutations aren't that old in the sense we have known about them for more than 10, 12 years, right?
There was one report from Israel about Parkinsonism in Gaucher patients from 1994, but it was buried in the literature. But it's really a 2004 New England Journal of Medicine that directed much more attention and helped us focus on GBA and GBA-PD.
And I can say, having been in the field back then, of course, we were nowhere near as advanced as the field when it came to therapies. And I know we often talk in the community about things will develop in 5-10 years. And quite rightly so, people who are affected by these disorders are disappointed that things haven't happened in 5-10 years. But I would argue in the last 10 years, or 14 years as is this case, is tremendous amount of development has taken place, and a lot of it driven through discoveries in the genetics of the diseases. But please continue.
Right. So I think that there are so many clinical trials using different approaches to treat GBA-PD or to address GBA-PD, from different types of interventions that I'm very hopeful that at least one of them would be successful and would really change the way we think about Parkinson's in general and GBA-PD. It basically would mean that anyone getting a Parkinson's diagnosis, part of the workup would be to get tested to see whether they're carriers of GBA or later on LRRK2, and would that mean that the treatment should be different? The field is so, I think people in the field, patients, people with Parkinson's, and the pharma are so excited about it that they're not letting they don't want to wait for us to develop the correct biomarkers and the complete clinical phenotype. They want to go ahead with interventions, but just like you mentioned, biomarkers that can trace GBA-PD and see how fast it progresses or how slowly it progresses and how to modify the progression are urgently needed. Mm-hmm. And these are needed in that all modalities are good for that. So it can be either biosamples, so spinal fluid, blood, or other products, or imaging, as mentioned before. But and this research on looking at biomarkers linked to Parkinson's is happening right now in parallel to the clinical trials.
Yeah, we should emphasize that that sort of research will accelerate the development of therapies tremendously if it's successful. And it may seem a little bit boring to measure things in people's blood and see if there's subtle changes, but it's tremendously important for the research community. So what do you think are the most urgent issues, you as a panel, the most urgent issues that are facing therapies that target GBA today? You know, if you were going to make a top 2 or 3 list, and Jonathan, if you want to chime in, if you unmute your phone, you can chime in. You sit in a special role as a funder and a founder of the foundation that plays a very important role in this area. Anybody can speak, but go ahead.
Looks like he may still be muted. This is Gaurav. I'll jump in while we figure that out. I think the number one issue today is probably finding patients for enrollment. As Roy was saying, I think in the future, it'll become common practice for any patient diagnosed with Parkinson's disease to go have a genotype performed, understand their mutation status. I think today it's still a struggle to find enough patients to run all of the trials that we have. And ultimately, it will be those clinical trials that help get us to the answers that we need to go to. So I'll start there as I think the number one issue facing the community today. And then I think number 2, just to expand on it, would be biomarkers. And I think ways to stratify patients around biomarkers, understand who's likely to progress, and what do the biomarkers mean with respect to their disease and their ultimately clinical status would be ultimately very important also.
So Ben, it touches a little bit upon what you said earlier at the other question. We talked about, what can one do if one finds out one has one of these genes? And maybe you want to expand on this, Ben, the notion of being an advocate and a participant.
Yes, and while it's important that we get as many people as possible into these trials, we also, I think, have to be thinking about the flip side, and that is the individual has a very difficult decision to make in many different ways. For a lot of different reasons, it's not an easy thing to just jump into a trial. There's a lot of barriers along the way that I think we could do a better job of facilitating, or at least breaking down the barrier of entry to. And also, it's a little bit confusing simply because there are different approaches being tried at the moment. And one doesn't have any good understanding, I guess, of which approach is going to be the most beneficial for them particularly. So it's hard to know exactly which of the ongoing trials would be best suited for each individual person that's thinking about diving into one of these trials.
So I could mention we were supposed to have an additional panelist on board today. Dr. Tony Shapira sends his apologies. He's traveling and he was hoping to chime in from an airport. That didn't work out technically, but he is running a trial where there is a drug that's being repurposed and being tested in people with GBA mutations. It's called Ambroxol. And maybe somebody wants to explain what kind of trial that is. So this is not a high-tech, complicated gene therapy trial, but it's actually a drug that's available in a pharmacy today. And how does that work, and how can you do this sort of thing?
Yeah, can you hear me okay?
Yes, we can, Jonathan. Welcome on.
Okay, so Ambroxol is an interesting drug because, it is in Europe easily available, and it is an activator of glucocerebrosidase. I think the concern people might have is that the dose, if you buy them online, you can buy them through Amazon. It's about, I think 75 milligrams per pill. So the lowest dose tried in some of the trials by Zaiwi and by Tony have been 1,200 milligrams. So you're talking about, I don't know what that is, 15 pills or something like that per day. So we need to find a better formulation. and we need to find a formulation that clearly crosses the blood-brain barrier more efficiently. But it is what's the most encouraging is it's something that people can do and not have to wait for a clinical trial where they might get a placebo arm where they'll have no assistance in their disease.
So I could mention that the trial that Tony's running is under the final analysis stages, and I think those results will be made more public soon. But I believe that they're sufficiently encouraging for there to be discussions about doing further trials with Ambroxol.
Yeah.
Gaurav, do you want to say?
I think I can say for everybody on the panel, we're rooting for them, and this is again, this will allow for a drug to be immediately on the market if it proves to be efficacious. The problem with some of the prior trials is they've not had a placebo arm, and they've typically had, I don't know if Tony's is this way, but have had, a low dose and a high dose, and it's hard to, and every patient's baseline was sort of the control. There's a little bit of a challenge to understand 100% of what we're looking at. But I look forward to seeing the full data set when it comes out soon.
Yeah, it will be very exciting when it's made public. I just want to interject a short question here that just came up from Keith Woolley online. He says, how does MSA differ from Parkinson's with respect to the inherited genes? Perhaps I can answer and say that we know much less about the genes that cause or affect MSA risk. And we know with Parkinson's at least 100 locations in the genome that affect the risk. And then there is a handful to 10 genes that clearly cause inherited familial Parkinson's with a much clearer inheritance. So we know a lot more about Parkinson's, and the overlap isn't that great, actually, which is surprising. It's hard to study MSA. There are so few cases, and the studies have been difficult to do. I don't know if somebody wants to give a more informed answer than that.
So the only one thing I wanted to add is that in addition to GBA that may be a risk for both, patients who die with alpha-synuclein mutations have alpha-synuclein deposition in nerve cells, just like Parkinson's, but they also have MSA-like changes in glia cells. So alpha-synuclein mutations is probably a risk factor for both MSA and PD changes in the brain.
And there is an additional complicating factor with MSA on top of what we just said. It's not a very homogeneous disease. We talk about Parkinson's being related to Lewy body dementia, but MSA has different variants and the pathology is different. So maybe they aren't all having the same genetic risk factors. But I want to wrap. Yes.
Sorry, going back a little bit, just covering some of the difficulties involved with recruitment in the trial. So speaking as a patient, and I think Ziv had at some point he wanted to mention as well regarding this, it's still hard to know how much an individual's mutation contributes to their actual Parkinson's disease. I can just say it's only one case, but this is a case of many as well. I have a history of this mutation in my family, but no history of the disease. So there's obviously more at play here, and it's hard to account for all the factors that go into this disease. But I think Ziv had a point he wanted to add on top of that as well.
Go ahead, Ziv.
Yeah, so there are some other difficulties on top of what was mentioned regarding GBA. Some of them are true to Parkinson's in general. I mentioned in the beginning mutations in GBA.
You are drifting out, Ziv. Can you get closer to the microphone, please?
Sure. Can you hear me well?
Now it's good. Thanks.
So there are different mutations, as I mentioned in the beginning, in GBA. And if you think about the Ambroxol trial that we just mentioned, this is a chaperone. It's a molecule that is taking the enzyme from one part of the cell into the lysosome. So of course, we don't know the results of the trial, but the way I see it, this drug can be useful for mutations that retain the protein in that part of the cell. There are different mutations that the problem that they cause is not that. There is no problem with the transport of GBA into the lysosome. So I think if Ambroxol will be useful, it will probably be useful for only specific types of mutations and not necessarily to all GBA mutations, because in some cases there's no protein at all. So even if you do give a chaperone, it's not going to help because there's no protein produced in the case of non-mutations. Now, on top of that, we have the problem in clinical trials, and that's true for all clinical trials that we do them on patients with, even if it's early Parkinson's disease, they already have some quite extensive damage that was done, and the cells that the drugs are target, many of them are already in the dead or in the process of dying, or at least the synapses are not functioning. So this is a major issue. We need to try to find a way to help us identify individuals in much earlier stages and identify specifically those that we know or we think that we know are going to develop Parkinson's, where the brain damage is much less extensive. And lastly, the last challenge that I want to mention is the heterogeneity. Even in GBA mutations, even carriers of the exact same mutation, and that's relating to what Ben mentioned, some of them will develop the disease, some will not. Some will develop it at an early age, some at a very late age, some will progress fast, some will progress slow, and we still don't know all the different factors that affect that. And if we want to do a successful trial, we want to be able to try to control for as many as possible of the variants that affect the rate of progression. Because if you have two arms for a trial, the treatment arm and the placebo arm, and you don't balance them in terms of how fast the patient progress, one arm can progress faster than the other or slower than the other just because of other factors, genetic factors, environmental factors, unrelated to the drug, which can lead to either false positive or false negative results, depends on where in which group. So these are many different challenges that we have, and there are actually many more. So this brings me always back to the notion that we also need to think about, we want to continue with the trials, obviously, and we want to try to progress, but we also should do a lot more basic science and a lot more to understand the different factors that affect the progression of GBA and Parkinson's.
So I want to tie into that with two of the questions from the audience. But before I do that, I want to go back to what you just said a minute or so ago, that we talk about precision medicine or personalized medicine, and we sort of think, okay, here's a drug that's going to be tried in a group of Parkinson patients with a certain mutation in a certain protein. But you're saying, not only do we have to have a group of patients with a mutation of GBA, but each mutation may be specific and need a specific therapy. So that is what we might then call super precision medicine. Now, the question that came up here, I think, is good, and anyone can chime in. It might be your question, Siv. What's your favorite hypothesis to explain how GBA affects synuclein? Is it lysosomal impairment with less degradation? Is it more cell-to-cell transmission of aggregates? Or are the lipids modified, so the lipid environment in the cell is modified? We know that the fats, the lipids in cells interact with synuclein. And the other question that's tied to this is from an anonymous person. The first one was from Elodie Angor, but the anonymous person says, how can you study this? Can you use iPS models? These are stem cells made into nerve cells. So a lot of things to answer there. What do you say, Sid? Do you want to start? Kick it off.
Sure. So this is obviously almost a guess, because we do know some things about GBA and alpha-synuclein and about the potential mechanisms, but we do not fully understand how GBA mutations lead to alpha-synuclein accumulation. My personal guess and things that we work on here at my lab, we think that GBA mutations affect the composition of the lysosomal membrane, which in turn affects its ability to perform autophagy. Autophagy is a process where the lysosome takes in synuclein to degrade it. And if we change the composition of the lysosomal membrane, this process can be affected. So we think that GBA mutations, the way that GBA works, it works on the membrane of the lysosome. So when there is reduced activity of GBA, the lysosomal membrane composition changes. So that, and then alpha-synuclein, that's what we hypothesize, is that alpha-synuclein cannot be properly internalized into the lysosome and properly degraded, and that leads to its accumulation, toxicity, and so on. But that's one out of many different hypotheses, and I think that maybe others would want to add to that.
Yes, go ahead. If anybody has a view on this, are stem cell models where you culture stem cells and make them into nerve cells, are they a good approach? You can take these cells, of course, from people with GBA mutations? Gaurav, Roy?
Sure, I can say a few.
Okay.
Roy, go ahead.
So I think that to answer the question of how to prove that what in GBA causes Parkinson's, I think that in order to convince everyone, one would need to show the link in more than one model. So you want to show, for example, let's say that you think that it's the reduced activity of the enzyme that leads to Parkinson's. You want to show that lower activity in people with GBA mutations, the lower your activity or higher the risk for Parkinson's or the faster the progression. And then you can do the same thing in IPS models where you can reduce the activity with inhibitors and see that the lower the activity, the higher the faster the aggregation of alpha-synuclein. So whenever one has a theory linking GBA to Parkinson's, this theory has to hold in at least in a couple of models, not just one. So it can be clinical model with measurements from people with Parkinson's who donated blood, urine, or spinal fluid. It needs to be in animal models of animals who have mutations in these genes, and it can be from cell cultures or it can be from IPS. But if something really links GBA to Parkinson's, the data should come from more than a single model.
Great answer. Gaurav, do you want to add anything to that?
No, that's pretty much what I was going to say. I will just add that I do think one of the most convincing models remains iPSC models. Particularly iPS-derived dopamine neurons. I think at least there you have some confidence that you're going to recapture the relevant biology in a patient with the relevant genetic background. I think looking at other types of cellular models like fibroblasts or even animals, it's hard to put as much faith into them. So I do think the iPSC model is quite powerful from the perspective of dissecting some of these key questions that are coming up.
So I think we're going to slowly move over to LRRK2, but before we do that, I'd like to ask a general question. How interested is the drug industry in GBA? And do they think that treatments that target GBA will be relevant for people who have Parkinson's but don't have GBA mutations? I'm
happy to take that.
Yes.
Based on the amount of phone calls we get, the pharmaceuticals are very interested in GBA PD. There are a bunch of companies, big ones, that announced their interest, including an ongoing trial of Genzyme Sanofi, Allergan and LTI. The Silverstein Foundation is funding a couple of clinical trials. There's Prevail. I think there's a true interest. I think the other thing that makes a lot of pharmaceuticals very interested is that I think there's convincing data that the GBA pathway is also important in idiopathic PD. Mm-hmm. So by now, 5 autopsy studies have shown reduced glucocerebrosidase, GCase, the enzyme encoded by GBA, in brains of people with Parkinson's, even without GBA mutations. So at least autopsy studies are convincingly showing that the GBA pathway is important in people even without mutations. And we have also shown a little bit of that in blood of people with Parkinson's without GBA mutations.
So a little bit related to that, Bruno Benitez has asked a question, are there variants in additional lysosomal genes that are associated with PDN? Maybe you want to fill in there, Roy.
I'll also absolutely have to have Ziv complement what I say, but I think the strongest association we see but for now is definitely with GBA. And the question that puzzles us, is the association the strongest with GBA because GBA mutations are so common and therefore easier to study? Or is there something specific about the GBA enzyme that is different from other lysosomal enzymes and enzymes that break down lipids? There are data about the enzyme involved in Niemann-Pick disease, acid sphingomyelinase. There are autopsies of people with Niemann-Pick type C, that is a different disease, that have alpha-synuclein deposits. When we measured enzymatic activities in people with Parkinson's without mutations, the association was stronger in alpha-galactosidase A, which is deficient in Fabry, not in Parkinson, in GB, not in Gaucher. So there is a lot of pieces of information linking other lysosomal genes and lysosomal enzymes to Parkinson's, but I still think that the strongest are with GBA for now. And maybe you can use it later as a segue, but in a way, some people think of LRRK2 as a lysosomal enzyme as well. So it's also a question of how do you define lysosomal enzymes.
And I think if one looks at the total... Oh, go ahead.
Sorry, yeah, I'll just add, I mean, I agree. You know, I might take it a step further and say, you know, maybe the relationship between GBA and PD is actually not special And there is potentially that relationship between general lysosomal insufficiency and Parkinson's disease and dementia with Lewy bodies. And just to add to the data set that Roy mentioned, in addition to the Niemann-Pick genes that have been associated, there's been findings with Sanfilippo, SALA disease, and NCL. And then I think the other one that comes to mind, just thinking about this relationship, is the fact that mutations in progranulin, which is a lysosomal disease, lysosomal gene, are affiliated with a neurodegenerative disorder as well. So I think clearly there's something, where there's smoke, there's fire, and clearly there's some deep relationship between these genes and these disorders.
And of course, the greatest risk factor of all for Parkinson's is aging, and we know there's a large body of literature to suggest that during aging, the lysosomal autophagy system shows some degree of decline. Do you want to add to that, Jonathon? I saw you went online.
Yeah, no, I was just going to say your original question was, is there interest in this disease from the pharmaceutical industry? And I think clearly there is. There are at least half a dozen GCase activators in development, multiple gene therapy approaches with different genes in development. And when you think about drug development, I remember many years ago, a few years ago, head of R&D telling me that if there was a genetic tie to the disease, they felt like they had about a 70% probability of success going from phase 2 to phase 3. But if there was no genetic tie, just strong phase 2 data, they had about the opposite, about a 30% probability. So here we clearly have a genetic tie, and we understand a little bit about what's going on. So the pharma industry also will be interested in this because it's a large orphan. So orphan drug status is given for diseases of 200,000 patients in the United States or less, where they're given an expedited review and additional help from the FDA. And this is, estimates are all below 200,000, but it's large enough where they don't have to charge an exorbitant price. So the pharma won't get the bad reputation of charging $400,000 for therapy. They can charge, for one and done therapy, theoretically on gene therapy, some fraction of that, and still make a good profit and keep the public happy at the same time, and the insurance companies.
And perhaps we should mention that to develop a drug, it's impossible to do it without partnering with industry. An academic scientist working in a university or a research institute can only make certain levels of discoveries, but doing the actual clinical trials is so expensive. There have to be investors who have the possibility of making a profit and can take that risk.
Yeah.
So it's very important.
Yeah, we are at the Silverstein Foundation, since we are, all of us are many of us are venture capitalists, and we work with some great advisors, like some of the people actually who are on this call. We're very focused on trying to fund things that can be sustainable, meaning be picked up by a large pharmaceutical company because the science is so interesting, get funded by venture capitalists. So for example, we started a company, Prevail Therapeutics, which has been mentioned a couple of times today. About 9 months after we got licenses and hired an initial team, they're able to raise $75 million from outside investors. So that's going to allow them to go through all of this year. It'll last them 3 years of cash and enough to get a proof of concept for the trial. So we're very excited about that, and that's really the way modus operandi for the Silverstein Foundation is create things that can be self-sustainable by either getting pharma excited or getting investors excited.
So I'd like at this point, which I think is a kind of optimistic point, hearing that there is a lot of interest in GBA mutations, I'd like then to move over to LOCK2. Before doing so, though, I see there are many questions we didn't have time to answer, and we'll try to send email answers to these. They'll be collected by folks at the Cure Parkinson's Trust, and we'll try to respond somehow by email to all of them. Now, as we go over to LOCK2, I'd like to ask the same question. This is a different protein and has many mutations that are associated with disease. So the question asked before with GBA, the same question for LOCK2 is, what is the role of LRRK2 in the development in Parkinson's? And will everyone who has a LRRK2 mutation actually develop Parkinson's disease? Who wants to answer that?
I can say a few words to get the conversation started. So
yes.
to the second question, LRRK2, like GBA, is a low penetrance mutation, so that probably not the majority of patients who harbor or carry mutations in LRRK2 will eventually develop Parkinson's disease. You know, like GBA, it's a relatively newly found gene, so we're still working out the exact risk affiliated with the disease. I do think, and this might be something that my other panelists might have views on as well, that the role that LRRK2 plays in the cell and the way it might contribute to Parkinson's disease is a little bit less well understood than GBA in some ways. For instance, I think with GBA, there's a sense of consensus that we need to increase the activity of GBA to help Parkinson's patients. With LRRK2, there may be reasons to investigate that a little bit more. And so whether you're trying to increase or decrease the activity clearly influences how you would approach a treatment. That being said, there are, just like GBA, there's LRRK2 inhibitors that are in clinical studies right now for Parkinson's disease. So some people think the best way to answer that question is to dive right into the human experiment and try to inhibit it with these, again, small molecule pills that we have. So there is interest into doing that, but you know, on this side of the table, as someone who works to invest in drug development, I do think that some of the scientific questions are a little bit earlier in our understanding.
Would the panelists share that view? I mean, some people might say we know quite a lot about LRRK2. Roy?
I think that our experience with LRRK2 show us how scientifically fortunate we are with GBA, because LRRK2 is way more complicated. Even the association with Parkinson's is more complicated. A lot of people who donated their brains and reached autopsy with LRRK2 mutations don't even have alpha-synuclein deposits in the brain. Some do, some don't. Some of those who don't have alpha-synuclein have tauopathy in the brain, as opposed to GBA, where the association with alpha-synuclein is much stronger and consistent among different studies. So first, we don't know exactly what LRRK2 does, and then we don't know exactly how to predict what kind of a pathology would LRRK2 lead to, so it makes potential interventions even more complicated. I think it sounds reasonable, and also just to, again, highlight how difficult it is with LRRK2. So when we found the association of GBA and Parkinson's, people already knew what GBA does. We know that when there's no GBA in the cell, in the body, Gaucher disease develops, lipids accumulate. There's decades of knowledge and treatment that is available for Gaucher mutations for GBA mutations from the Gaucher world. In LRRK2, it was much more complicated, and the gene also discovered in 2004. And only in the last couple of years we know that this gene is a kinase, and only in the last couple of years the substrates of the kinase were discovered. Previous to that, all we knew that LRRK2 phosphorylate was itself. So now that we have a better understanding of what are the biological substrates of LRRK2, I think that the research will catch up quickly. My guess is that it really is a gain of function and a toxic gain of function of the LRRK2 mutations. It's pretty convincing in G219S, the most common mutation in Ashkenazi Jews.
But could it be said, therefore, there's an advantage in the sense that it's easier to inhibit an enzyme than try to activate it?
Right. So pharmaceutical pharma knows how to produce kinase inhibitors. That's definitely an advantage. But even with GBA, there is one clinical trial for an inhibitor that inhibits the counter enzyme, the synthase that does the counter effect of the glucocerebrosidase. So, but this is true. So kinase inhibitors are easier to develop, and that's one advantage of LRRK2 research. The other is that probably the risk for Parkinson's in people with LRRK2 mutations is higher. So when you see someone with LRRK2 and Parkinson's, you're more convinced that they really have Parkinson's because of the LRRK2 mutation, where it is very possible that a lot of people with GBA mutations and Parkinson's have Parkinson's, and it may have not been because of the GBA mutation that they had.
Yeah, could you describe those numbers, because they're really interesting? So if you have a LRRK2 mutation, what is the likelihood that you're going to get the disease during your lifetime? I know it's a big ballpark.
No, it's very controversial, and it's very hard to study, because usually all those studies are done in LRRK2 is basically finding a lot of patients who have Parkinson's and LRRK2 and then ask them about their parents. And their parents lived in a different generation where Parkinson's was more difficult to diagnose, and many times people missed the diagnosis thinking that people were just stooped or shuffling their gait and didn't call it PD. Based on those family-based studies, the estimation are around 30% penetrance, so 30% to 50% of the people with LRRK2. 30% would be probably the closest name that we have in the literature now of people with LRRK2 mutations will develop Parkinson's disease. But it gets complicated with the question of what's Parkinson's disease. If we have a patient who had many years of symptoms, responded to Sinemet, and then passed away, and there's no alpha-synuclein in his brain, did he have Parkinson's disease by definition or not? So the penetrance estimations and calculations are complicated. I do want to emphasize that both with GBA and LRRK2, they're absolutely incomplete. I examined an 89-year-old who had both GBA and LRRK2 mutations, and to my exam was completely within normal range to what's expected at 89.
So a person who had 2 mutations in 2 different genes that can cause Parkinson's.
Right, and could still stand up from a sitting position with his arms folded at 89.
Does anybody want to add to this discussion about LRRK2 and the challenges, but also I think opportunities, because it's a druggable target?
I can add one more comment. I completely agree with what Roy said, and in many ways. We are very lucky in the GBA world, because it has been a well-studied biology because of the affiliation with Gaucher disease. In fact, one of the first ever biotech products was an enzyme replacement therapy for Gaucher disease, so the community is very comfortable with replacing it, and we're very clear that it's a loss of function. The one thing I'll mention about LRRK2 is that the inhibition strategy, you start to run the risk of hitting other sort of tissues outside the brain. So in the early set of experiences, when we as a pharmaceutical industry developed inhibitors for LRRK2, it seemed like they started to cause some issues in the lungs. Seems like we might have been able to engineer some of that out now, but one of the remaining concerns is whether we can specifically hit the abnormal activity of LRRK2 in the brain and spare the normal activity that seems to be present in things like the lungs and the kidneys. So just to add to the challenges and how it might be a little different from GBA and sort of a drug development perspective.
So LRRK2 obviously has an important role in the brain and in nerve cells, but could it have a role in the immune system? We know that the immune system and inflammation certainly is in one way or another involved in Parkinson's disease.
When we look at LRRK2 and GBA and try to look at similarities, so one of the similarities that is obvious and probably chance is that both are more common in people. Mutations in both are more common in people of Jewish ancestry. More biological commonalities is maybe the lysosome, but also the immune system. So when GBA mutations are deficient, when the glucocerebrosidase enzyme is deficient, the disease is a disease of the macrophages. And also with LRRK2, LRRK2 is highly expressed in macrophages. And in genome-wide association studies, LRRK2 variant, not the mutations linked to Parkinson's, have been associated with immune-related conditions like Crohn's disease. So I think that this opens a whole different webinar of the role of the immune system in Parkinson's, and what can we learn about the link between the immune system and Parkinson's from GBA and LRRK2.
Mm-hmm. So it could even be tissues that are outside the brain that are involved in the disease or the way it starts, potentially.
Absolutely.
Ben wants to say something about that. I know you have an interest.
Yeah, it's all fascinating. It all ties together in unique and interesting ways. I guess it's hard to know exactly. So there's an ongoing challenge, I guess, in the Parkinson's community to find out where everything kind of starts and where to pinpoint this kind of causal chain of destruction that takes place in some sense. There's all sorts of interesting theories going on, and I guess one thing to bear in mind is that each person's the most, or at least from my perspective anyways, the most likely scenario is that each person's kind of development of their disease might be unique in some sense to them, or at least they're part of a small cohort of people that develop this disease in a unique way simply because they're exposed to different things along their path that might help trigger this disease in the first place. And then some of these genetic mutations might come along afterwards and help kind of facilitate the development of this disease that then leads to some of the disorder that they experience. So each person's triggers and the things that enable the disease to carry forward might be, in some sense, unique to them, and identifying what those things are might be critical to understanding how each person develops the disease and then how to actually go about treating each person's, in some ways, unique case of this disease.
So at one point we talked about super precision medicine in terms of certain mutations in the GBA gene being different to other mutations, but it may also be that different individuals get the disease for some other additional factor that could be in the environment, I guess. And it makes it very complicated, no doubt. So before we wrap up, totally, we have about 3-4 minutes to go before the webinar closes. I'm looking if there are any new questions on LOCK2, and at the moment there aren't. But I would ask the panel to say something very quickly about other genetic causes of Parkinson's in relation to therapy. So Parkin mutations, when inherited from both the mother and father in an autosomal recessive way, can cause typically a young onset disease. Is there any hope there that there could be personalized precision medicine treatments for people with Parkin mutations?
I'm happy to comment on that, and I think I'm happy for the other panelists to join in as well. So definitely, I think that there is going to be a role for Parkin-directed therapies. The one challenge, I think, relates back to the earlier line of questioning around whether GBA therapies could work in the larger Parkinson's population, which I think there's good evidence to suggest it can. I think with Parkin mutations, that's a little bit less clear.
And by the way of LOCK2, we didn't really ask that question. And,
yeah.
You know, there's, as you know, an emerging literature saying that LOCK2 might be upregulated in idiopathic disease. That would be good because it would mean therapies that work in LOCK2 could be used in many patients.
I think LOCK2 is somewhere probably between GBA and Parkin on that spectrum. I think there's probably some controversy in the community around whether that's the case, but certainly there's boosters who think that LOCK2 therapies are likely to help patients with non-genetic forms of Parkinson's as well. And I think the jury's still out. I do think with Parkin, Parkin, just not is a basal off a molecule, so there's reasons to think that the therapy in non-mutant patients might not be helpful. Actually, the bigger challenge is that it's just such an exquisitely rare mutation that it's there's not near the same numbers of patients to really study, to consider enrolling a trial, and I think that's one of the bigger challenges now. And I think getting to the earlier point where in the future we might have just a reality where all Parkinson's patients have their genetic testing and counseling done, we might become aware of more Parkin mutant patients, but for now it's a pretty limited population. But nonetheless, we're aware of many efforts to develop therapies for them, and so I think, but they're earlier.
So, I mean, you're absolutely right, but I don't want us to sound too pessimistic when it comes to Parkin. I think everything you're saying is absolutely right, but there are companies interested in Parkin because of the pathways and its role in the mitochondria, the energy factories being pretty well defined. So
exactly.
Yeah, and I could just quickly add that a lot of these things tie into one another. There was a recent paper put out by Dmitry Krank's group showing the connections between the synapses, the mitochondria, and the lysosome. So in some ways, many of these pathways might intersect in some people, and targeting multiple different pathways might be the way to go for a lot of cases.
Yes, combination therapies, which might be the subject of a future webinar. We welcome any suggestions you have for topics you want us to discuss. we're actually at the point when we are supposed to wrap up. I really like to thank the panelists for a wonderful discussion. I think you've given a lot of insight and a very nuanced view of what's going on. I've learned a lot also during this seminar, so I've enjoyed it. And thank you for everybody who's listening in, and we hope to see you again in about 3 or 4 months from now. And once again, if you have any ideas about good topics, we are all ears and we'll be considering them. So thank you, everybody. And I think we're going to say bye-bye for now.
Thank you.
Thank you. Bye.
Thank you very much.